Okay, well, good morning. Thanks everyone for being here. I understand how precious our time is, so it really means a lot for you to spend the next couple hours with us. And really, I'm going to give you, over the next 90 minutes, give you the kind of the 20-year journey on the discovery of nitric oxide and how we've been able to translate these basic science discoveries into product technology that we're developing for drug therapies and perhaps most importantly, consumer products that have been proven safe and effective for restoration of nitric oxide. So thanks for the kind introduction, and I kind of gave you a background of kind of where I started, and now I'll show you where I'm headed. But when I started my own independent research career in academia, we were really focused on trying to answer 4 questions. How does the human body naturally produce nitric oxide? What goes wrong in people that can't make it? How do they present clinically? If you develop nitric oxide deficiency, how does this present clinically? And then most importantly, which I'll share with you towards the end, is how do we fix or restore endogenous nitric oxide production? Because hopefully I'll convince you that this is one of the most important molecules produced in the body of humans. And as you mentioned, this 20 years in academia and drug discovery, of dozens of issued patents, have published over 100 peer-reviewed papers in the scientific literature, and I've given over 300 lectures around the world. These are my conflicts of interest. We've commercialized and productized a lot of the discoveries. I'm going to tell you a little bit about our drug discovery program through Bryan Therapeutics, but this is just to inform you that obviously I have a conflict of interest. We've been able to very successful at commercializing some of the discoveries. But in academia and really in research and development, my job is to see what everyone else has seen, but to think what no one else has thought. And what I hope to share with you is kind of give you some of those original discoveries and original observations where we were able to see what everyone else had been thinking, but thought a little bit differently, and how this change in thought really changed the paradigm on how we deliver safe and effective nitric oxide. So obviously, I'm going to spend the majority of this time on nitric oxide, but as you're going to learn through this entire Real Truth About Health Summit, there are other important concepts in medicine and physiology that have to be addressed. So nitric oxide is foundational. It's extremely important, but I want to make it very clear. This is not a silver bullet. This is not an end-all, be-all, cure-all. You still have to address other root causes. And so broadly speaking, what I've learned in 25 years in academia is that people get sick for 2 reasons and 2 reasons only. Number 1, their body's exposed to something that's toxic. And number 2, the body's missing something that it needs. And whether it's a nutrient deficiency or exposure to a toxicant, the end result is disruption in nitric oxide production, decreased circulation, increased inflammation, oxidative stress, and immune dysfunction. And that bottom sentence explains every single chronic disease, whether it's diabetes, cardiovascular disease, Alzheimer's, autoimmune disease, liver disease, lung disease, neurological disease. It's decreased blood flow, increased inflammation, oxidative stress, and immune dysfunction. And I'm going to show you how to get to the root cause of all of those. So first, let me just start out, because I like to start out with broad concepts and kind of lead you on this kind of delving deeper into the underlying biochemistry and physiology. But the one thing that I like to start with is look at important clinical observations. And then if we can take important clinical observations and work backwards and figure out what's leading to that clinical presentation, then and only then can we develop safe and effective product technology. And so this is the work of Daniel Amen. Dr. Amen has one of the largest databases of SPECT scans in the world on his patients. And a SPECT scan tells us how well perfused an organ is. And in this case, it's the brain. So on the left, you can see a healthy brain, and this nicely contoured SPECT image tells us that all regions of the brain are getting sufficient blood supply, adequately perfused, and obviously this patient doesn't have any neurological deficits or neurological problems. If you look on the right in the SPECT scan of an Alzheimer's brain, you can see that's completely different than a healthy brain. And so these holes in the brain are areas in the brain in which there's decreased perfusion, and we call this focal ischemia. Select parts of the brain that aren't getting adequate blood supply. So what happens? Well, the brain atrophies. We don't get the good stuff in. We don't take out the waste products. You get misfolding of protein. You get insulin resistance, type 3 diabetes. And those are the underlying hallmarks of Alzheimer's disease. So, and then if you look in an angiogram, obviously this is an invasive type of imaging. And in this patient, you can see here in the angiogram here that there's a thrombus or an occlusion in the left circumflex. So this patient presented with a heart attack, shortness of breath, chest pain. Fortunately, he was taken to the cath lab and reperfused. They aspirated this thrombus in the left circumflex. And you can see here completely restored coronary flow. And if you do this in a timely manner, you can completely preserve the function of that organ. So in this patient here, even though he suffered a heart attack, complete occlusion of the left circumflex, when we restored blood flow to that organ, he came out with normal cardiac function. No necrosis, no myocardial infarction, normal cardiac contractility, and normal function. And then if you dig a little bit deeper and look at neurons, whether they're sensory or motor neurons, these are tissues and cells that have to have adequate blood supply. So these neurons are typically very well perfused because we have to deliver oxygen, nutrients, sodium, potassium to these neurons so they can initiate an action potential to conduct nerve transmission. But when you develop microvascular disease and these small blood vessels become chronically constricted, then these neurons atrophy. And this is the basis for peripheral neuropathy. So the whole point of taking these three examples, the brain, the heart, and nerve tissue, is that every single chronic disease is characterized by decreased blood flow to that affected organ. But if I showed you, if you can restore blood flow, you can correct many of these chronic conditions. So the next question is, how do we do this? And that's the role of nitric oxide. And so nitric oxide is a gas. It's naturally produced in the body. And once it's produced, it's gone in less than a second. But during that second it's around, it activates a number of cell signaling pathways that then start these second messenger systems that lead to a number of important biological and physiological effects. It's a free radical, meaning that it has an unpaired electron, but because of the polarity of this NO bond, nitric oxide is actually an electron donor. And any electron donating species is antioxidant, anti-inflammatory by nature. So that's a very important kind of a physical chemistry attribute of nitric oxide. And I like this quote by Val Fuster, who was famously quoted in 1998 that the discovery of nitric oxide and its function is one of the most important in the history of cardiovascular medicine. Now, fast forward 26 years, and that statement still holds true today. And I show you this seemingly very complicated slide here, but this is, again, one of these papers and publications and discoveries that completely changed the way we thought about nitric oxide. And this is from my good friend and colleague, Jonathan Stamler. But what he discovered, and he describes in this research study here, is that nitric oxide is required for oxygen uptake and oxygen delivery. So the cardiopulmonary cycle is a three-gas system. We breathe in oxygen, and we deliver that oxygen from the arteries to the veins and the capillaries, and then we pick up carbon dioxide, which is a metabolic waste product, and then we exhale that carbon dioxide in our exhaled breath. But this doesn't work unless we have that third gas, nitric oxide, bound to a cysteine residue on hemoglobin. So, and we saw this over the past 4 years, people with respiratory viral infections develop hypoxemia, right? So their blood oxygen saturation would go from a normal of 98 to 99 to, in some cases, high 70s, low 80s. And this is critical hypoxemia. So what do? We put them in the hospital. We give them 100% oxygen. That doesn't improve their oxygen saturation. So you put them on a mechanical vent. That still doesn't do it. And many patients died. But what we're finding is that if we could have just restored nitric oxide, which is what we did in our nitric oxide drug study, you can restore the functionality of nitric oxide. You can see blood oxygen saturation go from 78 to 96 within 8 minutes. And that's just breathing room air. So the problem is in these patients with COPD, emphysema, respiratory viruses is they're not lacking oxygen. They've lost the ability to take up oxygen and deliver oxygen because they've lost the ability to produce nitric oxide. So I'll come back to that at the end, but that's a very important concept when we look at tissue oxygenation and delivering oxygen to the tissue. But it's involved in basically every biological function. It's what controls gut motility in the periphery. I'm going to spend a little bit of time talking about the science around nitric oxide mobilizing stem cells, leading to regeneration, repairing, and replacing dysfunctional tissues. It's a neurotransmitter in the central nervous system, controls cell cycles. So in patients with cancer and metastatic disease, nitric oxide is critically important. It's a bronchodilator as well as a vasodilator. It's the molecule responsible for erections in both men and women. And it's part of our innate immune system. It's how our immune cells kill off invading pathogens from viruses to bacteria. So people who can't make nitric oxide, when you lose the ability to produce nitric oxide, you get multisystemic disease. You become immunocompromised. You get high blood pressure, erectile dysfunction, gastroparesis, advanced aging, sometimes metastatic disease. And the discovery of nitric oxide was deemed so important that in 1998, the Nobel Assembly awarded a Nobel Prize to Bob Furchtgott, Lou Ignarro, and Fred Murad for the discoveries for nitric oxide as a signaling molecule in the cardiovascular system. And these were kind of the leaders in the field, and I've been very fortunate to work and train under these Nobel laureates. As you mentioned, Fred recruited me to join the faculty of UT Medical School in Houston back in 2005. And Lou Ignarro remains a close friend and colleague and has written the foreword to several of my textbooks. But again, I'm going to start broadly because I think what we're all interested in this Real Truth About Health Summit is how do we live longer, but more importantly, how do we live better, free of disease? And this emerging field of longevity has really opened up therapeutic potential for addressing the root cause of aging. So there's three objective measures of aging. There's telomeres, there's mitochondrial dysfunction, so every age-related disease is associated with mitochondrial dysfunction, and then there's loss of stem cell function and repair. And I presented this concept over a decade ago that nitric oxide is the unified theory of aging because it's nitric oxide that controls and regulates telomerase activity, preventing telomere shortening. Nitric oxide is what induces mitochondrial biogenesis, improves ATP production, and nitric oxide is the signal that tells our own stem cells to mobilize and differentiate. So really quickly, I'm just going to take you through all these because I don't want you to take my word for it. I want to present to you the published peer-reviewed science. And so telomeres, as I mentioned, are the very ends of the chromosomes that with each cellular replication, these telomeres can become shorter. And so now it's known that shorter telomeres, shorter lifespan. If we can prevent telomere shortening, we can prevent early death, early demise. So this is a very rich area of science dating back to the late 90s. So where does nitric oxide come to play in this? Well, it was first discovered in the early 2000s that nitric oxide is what activates the enzyme that prevents telomere shortening. And not only is it affecting the function of the telomerase enzyme, it actually controls and regulates the genetic expression and translation of that enzyme. And as you'll see here in 2009, the very important discovery here that nitric oxide is the master signaling molecule in the regulation of telomerase activity and provides the opportunity for innovative therapeutic approaches based on using nitric oxide active compounds. And here's the kind of the co-expression of the eNOS enzyme with the estrogen receptor alpha. And so this co-localization that allows for the genetic expression of the telomerase enzyme. But the point I want to make, and I'm going to introduce this concept later on, this requires a functional NOS enzyme. This is the enzyme, nitric oxide synthase, that produces nitric oxide. So if this enzyme isn't coupled and functional, then not only do you not get the expression and the translation of the telomerase enzyme, even if it's expressed, it can't be active. So loss of nitric oxide leads to loss of telomerase activity and shorter telomeres. And then what about the mitochondria? The mitochondria are the energy-producing organelles of every cell in the body. Lower numbers of mitochondria, mitochondrial dysfunction are hallmarks of all age-related chronic disease. And it's nitric oxide that controls ATP production. That's the energy currency of the cell. It actually regulates oxidative stress and the production of reactive oxygen species. It induces apoptosis. And as I'll show you, it's the signal that tells the cell to produce more mitochondria. So it controls everything we know about the bioenergetics and the metabolism and energy production within the cell. So again, we start with important clinical observations. Everybody knows that moderate physical exercise is cardioprotective. Everybody knows that caloric restriction, intermittent fasting, certain dietary pathways are cardioprotective, cytoprotective. But these mechanisms and observations are dependent upon the cell's ability to produce nitric oxide. So when we start to exercise or we do caloric restriction, that activates the nitric oxide production in the cell, leads to cyclic GMP. this produces an activation of what's called PGC1 alpha, and then that induces mitochondrial biogenesis. And this is the pathway of sirtuins, NAD. Everything that promotes longevity is activating this PGC1 alpha, which is dependent upon the cell's ability to produce nitric oxide. If you develop endothelial dysfunction and have an uncoupled NOS, you can fast for 3 days, 40 days, whatever. You're never going to get induction of this pathway until we restore the production of nitric oxide. And it's extremely important in performance, and it's performance on the athletic field, it's performance in the boardroom in terms of cognition, and it's performance in the bedroom in maintaining normal erectile function. So mechanistically, we know that nitric oxide is what extends the oxygen gradient into tissue. This nitric oxide is a reversible inhibitor of cytochrome C oxidase, so it allows oxygen to diffuse further into tissue by occupying that binding spot that oxygen would normally bind to. And then we know that at complex 1 and complex 4, it actually inhibits superoxide production and electron leakage across the electron transport chain, thereby mitigating the oxidative stress, recoupling the electron transport chain, and leading to more efficient ATP production with less oxygen. So the point here is if your body can make sufficient nitric oxide, then this what sets the stage for performance. And again, it's not just athletic performance, it's performance by every definition we define in physiology and medicine. And then what about stem cells? So we're constantly wearing ourselves out. Aging is just the inability to repair and replace dysfunctional cells. And 20 years ago, it was once thought that the older we get, the less stem cells we have on board. So that's what leads to age-related disease. Now we know that there's an enormous reservoir of stem cells in our fat, in our adipose tissue. And most people accumulate more fat with age. So it's not a loss of stem cell availability. It's the loss of the signal to mobilize our own stem cells and to mobilize and differentiate and repair and replace dysfunctional cells. And again, I show you the original research in 2003. It was revealed for the first time that nitric oxide is the signal that tells our own stem cells to mobilize and differentiate. And then the work we published on shows that the bioavailability of nitric oxide in patients may predict stem cell therapy success or failure. So again, if you lose the ability to produce nitric oxide, you no longer mobilize stem cells. And that's why the older you get, the less you repair and recovery from an injury or from exercise. So broadly speaking, you need nitric oxide to control all aspects of aging, prevent telomere shortening, increase mitochondrial biogenesis, mitochondrial efficiency, and mobilize our own stem cells. But now let's look specifically at vascular biology. And this quote here by this English physician over 400 years ago really leads to what we know about vascular biology. So the older we get, and many of you do this through carotid intima media thickness and look at the intima hyperplasia here, but in many cases, you start to see plaque deposition under the lining of the blood vessel. This leads to an obstruction, or we call stenosis, of the blood vessels. But it's really not the overlying stenosis and obstruction in patients with coronary disease. It's the stability of the plaque. So when this underlying plaque in the intima becomes unstable, and we call this soft vulnerable plaque, and this plaque ruptures, that's what leads to acute myocardial infarction, sudden cardiac death, if it happens in the coronary arteries, or ischemic stroke, if it happens in the cerebral arteries. So today we understand this entire process of onset and progression of cardiovascular disease, plaque formation, plaque instability, and plaque rupture. And it all starts with the functional production of nitric oxide in the lining of the blood vessels. So as long as your endothelial cells, and these are the single layer of cells that line all blood vessels throughout the body, as long as they can produce sufficient nitric oxide, then you don't get the inflammation, oxidative stress, and immune dysfunction that leads to the onset and progression of cardiovascular disease. And in fact, the functional loss of nitric oxide precedes these structural changes by many years. And many times it's decades. So if you already have an increase in the carotid intima media thickness and overt disease, that tells us that you've been nitric oxide deficient for many, many years. But there's good news. If we can maintain normal nitric oxide production, then we can prevent this. But perhaps more importantly and therapeutically, if you have the advanced progression of disease, we're seeing now plaque regression. So we can actually reverse cardiovascular disease, stabilize unstable plaque, and lead to regression of this disease. And we can do this by focusing on a single molecule, nitric oxide. So I'm trained as a biochemist and physiologist, so we don't just take important clinical observations, but we have to understand mechanism. And it's the inflammation, oxidative stress, and immune dysfunction that causes arteries to age and what leads to cardiovascular disease, which remains the #1 killer of men and women worldwide. And I'm going to take you through this, the original data showing that nitric oxide, increasing nitric oxide addresses all these issues around arterial aging. Let's start with oxidative stress. There are three cellular sources, three primary sources of oxidative stress, and this is superoxide and hydroxyl radical, these toxic, damaging oxygen radicals. And so here are the data showing that nitric oxide suppresses NADPH oxidase by post-translationally modifying this protein. Nitric oxide recouples the electron transport chain and inhibits superoxide production. And then in our paper in 2009, the enzyme that actually makes nitric oxide, if it's not coupled and functional, actually produces superoxide. So we know how to recouple NOS enzyme, prevent superoxide production, normalize nitric oxide production, which then, by the way, shuts down NADPH oxidase, shuts down the electron leakage and superoxide production from the electron transport chain. So focusing on restoring the production of nitric oxide addresses the root cause of oxidative stress. It's self-fixing. And the reaction, we know the kinetics of nitric oxide reacting. So nitric oxide not only suppresses and inhibits superoxide production, but even if there's always a little bit of superoxide being produced. And the only way to stop a radical reaction is through another radical. So nitric oxide binds to superoxide through this radical reaction, neutralizes this, and forms a cage-like molecule we call peroxynitrite. And then this is a cage-like molecule that simply rearranges to stabilize the electrons and forms inorganic nitrate. So you can see NO3 minus is the same as OONO minus. We also have enzymatic systems, thioredoxin, glutathione peroxidase, that detoxifies enzymatically this peroxynitrite into inorganic nitrite, which is a cardioprotective, cytoprotective molecule. So the whole point I want to make here is mechanistically, we know everything we know about nitric oxide and can understand the reaction kinetics and how it's working in an antioxidant and anti-inflammatory manner. So now I want to dig down into really the research we've done over the past 20 years, because before we can start to understand the mechanism of disease, we have to understand how does the human body produce nitric oxide? And I'm going to go a little bit into detail here because I think it's very important that each of you understand what pathways lead to nitric oxide production and then what you may be doing that's inhibiting these pathways. So the first pathway to be discovered was through an enzyme called nitric oxide synthase. You've heard me talk about this already. But this enzyme converts L-arginine, which is an amino acid, into nitric oxide. And this is based on the function of the enzyme. And then we also can improve nitric oxide from the diet based on the foods we eat, our dietary patterns, and as I'll show you, the presence of an oral bacteria and an oral microbiome. And then this pathway can create a source of nitric oxide. So one can compensate for the other. So if you eat a poor diet, but you have good endothelial function, then you really don't get into problems. And if you have endothelial dysfunction, but you can eat a good, healthy diet, then you really don't see many problems. But when you develop endothelial dysfunction, combine that with a Western diet, the standard American diet, it leads to complete nitric oxide deficiency, and we start to see onset of symptoms and the rapid progression of disease. So let me show you how this works in a normal, healthy individual. So this would be the cross-section of a blood vessel, and this red or pink here would be the lumen of the blood vessel. And the lining of these blood vessels are endothelial cells. This is the inside of the body, or the endothelium. And then surrounding every blood vessel is a layer of smooth muscle. And when smooth muscle contracts, it constricts the blood vessel. When smooth muscle relaxes, it dilates the blood vessel. So when we start to exercise, that's why it produces nitric oxide, and that's why exercise is medicine. It turns on this enzyme. As long as this enzyme is functional, converts arginine to nitric oxide, And nitric oxide is a gas. So part of it diffuses into the lumen, part of it diffuses into the smooth muscle. And there, in less than a second, it binds to an enzyme called guanylyl cyclase. And then this enzyme converts GTP into cyclic GMP. And cyclic GMP is a second messenger that activates calcium release, leads to smooth muscle relaxation and vasodilation. And that's why nitric oxide is a vasodilator, because it activates the second messenger, cyclic GMP, smooth muscle relaxation, and vasodilation. But interestingly, this is where drugs like PDE5 inhibitors, these are phosphodiesterase isoform 5 inhibitors. So in a normal active cell, you see an increase in cyclic GMP, activates calcium mobilization, and then this phosphodiesterase enzyme breaks down cyclic GMP. But if you can prevent the breakdown of cyclic GMP by using drugs like Viagra, you can potentiate the vasodilatory effects. And that's why these drugs are effective at improving erectile function in men, is because they dilate the blood vessels and they potentiate the vasodilatory effects of nitric oxide. And that's also the reason you're warned against a 4-hour erection, because you turn the signal off by the phosphodiesterase enzyme. If you inhibit that enzyme, then nitric oxide turns this switch on, and these PDE5 inhibitors keep it on. So you have to worry about an unsafe drop in blood pressure, 4-hour erections, priapism, and necrosis of that organ. So this is a very serious condition. But interestingly, these drugs have been on the market for more than 25 years now, and clinically, 50% of the men that are prescribed drugs like Viagra, Cialis for erectile dysfunction or urinary symptoms of BPH don't respond. So they don't get better. And why is that? And it's because in these responders, they're not making enough nitric oxide here to activate the second messenger system. So without nitric oxide, there's no cyclic GMP. Without cyclic GMP, PDE5 inhibitors can't work or potentiate the vasodilatory effects. So erectile dysfunction is a symptom of nitric oxide deficiency and the first sign of endothelial dysfunction. If we can restore nitric oxide production, we can overcome erectile dysfunction, and we can actually take non-responders to PDE5 inhibition therapy and make them responders. And then more importantly, you can lower the dose of these drugs, making them safer and yet more effective. So what happens with this enzyme with age? And it's the dysfunctional enzyme that occurs the older we get. And there are a number of contributing factors to this. But these are 4 independent studies that look at the age-related loss of nitric oxide production through this enzyme. So we lose about 10 to 12% per decade. By the time we're 40 years old, we only have about 50% of the nitric oxide we had when we were younger. And this is the age that people start to feel the effects of aging. 50% of the men over the age of 40 self-report erectile dysfunction. 50%. And most men aren't going to self-report erectile dysfunction. So I think the numbers are much higher. But today we know that we can prevent this age-related decline, or we can accelerate it if you live the Western lifestyle. Today we see 20, 25-year-old kids that have the vascular age of a 60 or 70-year-old because of their diet, because of their lifestyle. And then we see 60, 70-year-old patients that have the biological age, of a 30 or 40-year-old. I turned 50 last November, but I have the biological age of a 32-year-old. So we now know how to shift this curve to the left and to the right and hopefully prevent the age-related decline in nitric oxide production. And here are the data that show you. So in this particular study, we take well-trained athletes, we put them on an exercise treadmill, we increase their workload, we start an IV and just pull peripheral blood. And the amount of nitrite that shows up in the plasma, which is an acute phase marker for nitric oxide production, predicts exercise capacity. And this study was published by my good friends and colleagues in Germany at Heinrich Heine University, showing that exercise capacity is predicted by the ability to produce nitric oxide. Now, if you do that same experiment in 50 to 60-year-old patients with known risk factors for cardiovascular disease, they failed to increase nitric oxide production in response to exercise. And so what happens, they fail on exercise stress test. If you can't produce nitric oxide to dilate the coronary arteries upon increasing workload, then you can't increase the perfusion or oxygenation of the myocardium, and then you start to see changes in electrical activity, and this is now diagnostic for ischemic heart disease and endothelial dysfunction. So let me just get into the biochemistry of this enzyme, nitric oxide synthase. To convert arginine to nitric oxide, this is a 5-electron multi-step oxidation reaction. And it's one of the few, if not only, enzymes that requires 8 different cofactors and substrates. So number 1, this enzyme must be functional. It must be coupled. Number 2, we must have a sufficient pool of reduced tetrahydrobiopterin, glutathione, sufficient oxygen, magnesium. 75% of Americans are deficient in magnesium. NADPH, which people with G6PD deficiency have a deficiency of NADPH, FAD, flavin-dependent cofactors, and then heme iron. A lot of people are anemic. So all of this has to happen at the right time in the right space for nitric oxide to be produced. And the nitric oxide synthase enzyme is 2 twins. We call a homodimer. And these 2 twins that come together allow for this sufficient flow of electrons to convert arginine into nitric oxide. And then we get citrulline as a byproduct. But if we have oxidative stress and BH4 becomes oxidized, this enzyme becomes uncoupled, and now we reduce molecular oxygen to superoxide instead of nitric oxide. So it's the oxidation of BH4 that leads to NOS uncoupling and is the rate-limiting step in nitric oxide production. So we have to be able to preserve the electrical potential of BH4 to BH2 ratios. And most people don't understand this. Obviously, you have to be trained in physical chemistry and biochemistry to understand this. And for every biomolecule, there's an electrical potential. So electrons are negatively charged compounds or molecules or subparticles. And so there's an electrical potential for every biomolecule that's needed to extract an electron from that. And every molecule has a different redox potential. If you don't understand this, then you cannot prevent NOS uncoupling and prevent oxidative stress. So the reason I tell you this is, nitric oxide was once considered a miracle molecule. And miracle implies that we don't have an explanation, that it just happens. We don't have a scientific explanation. Well, that was 30 years ago. Today, there's nothing miracle about nitric oxide because we understand mechanistically how it's produced, where it goes, what it becomes, how it signals, and we know how to control it. So it was once thought, well, if you want to improve nitric oxide production, let's give L-arginine. And as many of you are aware, there are dozens, if not hundreds, of L-arginine products on the market targeted towards nitric oxide. But here's what happens if you don't understand the underlying enzymopathy in the patients with endothelial dysfunction. In this study in 2006, they took patients who had just suffered a heart attack and had compromised heart function, so an ejection fraction of, let's say, certainly less than optimal, less than 50%. Then they gave them arginine to try to improve vascular stiffness, improve contractility. They gave the patients the arginine. It caused more death than the placebo. So arginine, when added to standard post-infarct therapy, does not improve vascular stiffness measurements or ejection fraction and is associated with higher post-infarct mortality. Arginine should not be recommended following acute MI. Okay, very important study. They did the same thing with peripheral artery disease in patients and found that if you give arginine to patients with peripheral artery disease, they actually get worse. Intermittent claudication gets worse. So the conclusions of this study was arginine is not useful in patients with intermittent claudication and peripheral artery disease. And why is that? It's because in these patients, the NOS enzyme is uncoupled. They're not deficient in L-arginine. They've lost the ability to utilize that amino acid to make nitric oxide. And when you give arginine to an uncoupled NOS, you create superoxide and you exacerbate the underlying condition. So arginine should not be used in patients with endothelial dysfunction. So now let me take you back almost 20 years ago. I was a professor of molecular medicine at the Institute of Molecular Medicine at the UT Medical School in Houston, in Texas. We asked some very simple questions and try to answer this through designing rational experiments. So our whole thought process was, can we prevent injury from heart attack if we increase nitric oxide production in the heart? Because many of you know, if you're physicians or consumers or patients, you probably know a family member who had sudden cardiac death. Their first sign and symptom of heart disease was death. And then we know people who have had a heart attack, a widowmaker, high up in the LAD. They get to the hospital, they reperfuse, and not only do they survive, but they thrive, and their heart is actually protected. They have normal ejection fraction post-MI. So what is it about a heart attack in one patient that leads to sudden cardiac death? That same heart attack in the same spot of the coronary arteries leads to mild symptoms, but obviously taking action, going and getting reperfused, and there's no injury to the heart. Can we recapitulate this in animal models, and can we trace this phenotype biochemically? So this is a study from 2006, and my colleagues, David Leffer, they created a transgenic mouse where they overexpressed the nitric oxide synthase only in the cardiac myocytes. So we want to increase nitric oxide production only in the heart. And then if you occlude the LAD, so in these studies, we do open heart surgery, we tie a ligature, a suture around the LAD, the coronary artery, for 1 hour. So we cause a heart attack for 1 hour, then we cut the ligature, the suture, and then reperfuse that heart for 24 hours. And then we can do echo on these mice looking at cardiac function, contractility. Then we harvest the heart, do histology, and look at the amount of infarct or injury in the heart. In this paper published in 2006, the mice that have sufficient nitric oxide or overexpression of nitric oxide in the heart don't see any injury from a heart attack. Very important clinical observation. So we started taking the blood of these mice and trying to understand what is it about these mice that confer that cardioprotection. And then we harvested the liver and found that the liver biochemistry looked like that of the heart. And just to show we had a clean transfection only in the heart, not the liver, you can see the Western blot here. There's no overexpression of the enzyme in the liver. And understand that nitric oxide produced in the heart is gone in less than a second. So it will not survive transit from the heart to the liver. But yet something is modifying the liver biochemistry. And so the next question was, what does this mean, if anything? So we cut off the blood supply to the liver for 1 hour. We reperfuse that liver for 5 hours. And then we look at liver enzymes. And these are transaminase enzymes that are reflective of liver injury. So higher enzyme levels, higher liver injury. Lower enzyme, less injury to the liver. So the nitric oxide that was produced in the heart led to protection of ischemia perfusion injury in the liver. And we published this paper in the Proceedings of the National Academy of Science in 2008, showing for the very first time that nitric oxide was a hormone. What's a hormone? It's a molecule produced in an endocrine gland that has systemic effects, like testosterone produced in the testes of men or estrogen produced in the ovaries of women. It's secreted from those organs, but it has systemic effects. Nitric oxide is no different. It's secreted in the organ, the endocrine glands, the endocrine cells, or the endothelial cells. It has systemic effects. And this was my eureka moment in science and my epiphany, because this told me all we have to do is produce nitric oxide in a single biological compartment. Obviously, we don't want to induce genetic manipulation or gene insertions into humans. What if we could generate nitric oxide gas in the oral cavity? It's absorbed and it goes systemic. And that's been the basis for everything we've done going forward. So then understanding that all we had to do, and in these cardiac-specific nitric oxide mice, we saw an increased levels of plasma levels of nitrite and nitrate. So then we actually designed this experiment in mice because it's very easy to control the diet in mice. It's very difficult, if not impossible, to control the diet in humans. So we put our mice on a Western diet, what we call the standard American diet, or the SAD diet. And then we basically could calculate how much we took away, and then we gave this back in the drinking water. So we could use diet as a way to modulate steady state levels of plasma and cardiac levels of nitrite and nitrate. And then again, we subject these mice to heart attack, 1 hour, no flow ischemia, 24-hour reperfusion. You can see the mice that were on the standard American diet, about 40% greater injury to the heart. And about 50% of these mice didn't survive out to 24 hours. So the mice on the standard American diet, we give them a heart attack, 50% died. The 50% that survived had about 40% greater infarct in the heart. So compromised contractility. And if we let these mice live, they'll eventually develop heart failure and die weeks or months later. But if we give this back in the drinking water and replete what we took out from the diet, 100% survive out to 24 hours and about 30% less infarct in the heart than the control animals. So improvement in mortality and improvement in protecting the contractility and the function of the heart. Now we thought, let's push the envelope. Let's take mice that can't make nitric oxide because we deleted the gene that makes nitric oxide, and we put them on a Western diet. And this is the typical American, right? The typical American that has endothelial dysfunction that eats a Western diet. And you can see here, we can actually completely modulate these steady state levels of nitrite nitrate simply by giving an eNOS deficient mice a standard diet or putting them on an American diet and then giving this back in the drinking water. And here's a cross-section of the heart. The eNOS deficient mice, you can see here, the white tissue in this cross-section of the heart is dead tissue. This is infarct, this is necrosis, there's no contractility function of this heart. If we give it back in the drinking water, you can see extreme protection, about 60% less infarct, simply by giving a supplemental dose of nitric oxide, or in this case, nitrite or nitrate. So that told us that we can use diet as a first-line intervention for patients that are at risk for heart attack and stroke, improve vasodilation, stabilize the plaque, and if they happen to have a heart attack, we can improve outcome and maintain the contractility and the function of the heart. Well, what about diabetes? You know, diabetes is a worldwide epidemic now. It's not just a westernized disease. We've known for many, many years that once you get diabetes,
Dr. Bryan, it seems like your audio was cut out.
so I'll continue and hopefully you can hear me.
Now I can.
Okay, so people typically don't die from diabetes. They'll die from the cardiovascular complications of diabetes, heart attack, stroke. And so when we knock out the gene that makes nitric oxide, the first phenotype is insulin-resistant type 2 diabetes. So these eNOS deficient mice develop diabetes, completely recapitulating human disease. So this suggested to me that if you become deficient or compromised in the ability to make nitric oxide, that leads to insulin resistance. If you don't correct it, that leads to diabetes. Once you get diabetes, it further suppresses nitric oxide production. So there's this feed-forward mechanism that you have to restore the production of nitric oxide in order to intervene and intercede into the progression of diabetes. And this is a complex slide, but I show you this because this is where we tried to interrogate the insulin signaling to try to figure out where does nitric oxide play a role in glucose uptake in managing hyperglycemia. And so for some of you, this may be very complicated, but just bear with me here. So when insulin is secreted from the beta cells of the pancreas, it binds to the insulin receptors. And through this transmembrane receptors, it activates some intracellular signaling. PI3 kinase, AMP kinase, which by the way is the target of metformin, the primary anti-diabetic drug. But this is where the literature led us astray because we knew what happened here, but the terminal step was glucose uptake through GLUT4 translocation. But we knew that AMP kinase and PI3 AKT kinase converged on the eNOS enzyme. And this leads to a phosphorylation of serine residues, turns the enzyme on, and produces nitric oxide. But we knew in diabetics that this enzyme didn't work. So you can activate it, stimulate it, but if it isn't coupled, it can't produce nitric oxide. So then what happens to glucose uptake? And we engineered some cells where we tagged green fluorescent protein to the GLUT4 transporter. So we can actually visualize in real time the GLUT4, or the green fluorescent protein, which is tagged to the GLUT4, go to the membrane when we activate it. So we can activate GLUT4 translocation and improve glucose uptake simply by giving nitric oxide or nitric oxide donor drugs. And we published this 10 years ago. Just like in hemoglobin, there's two cysteine residues that are conserved throughout evolution that when post-translationally modified, that's necessary and sufficient for GLUT4 translocation, enhancing glucose uptake. But if the cell can't make nitric oxide, GLUT4 never gets the signal. Just like our stem cells never get the signal because there's no nitric oxide, GLUT4 doesn't get the signal to go to the membrane and bring in glucose. So what happens? We get hyperglycemia. That tells our pancreas, hey, I need more insulin. Then we get hyperinsulinemia, hyperglycemia. That starts this entire inflammatory cascade that leads to diabetic neuropathy, diabetic macular degeneration, retinopathy. Everything we know about diabetes and metabolic disease can be explained by a loss of nitric oxide production.